A periodic narrative of the evolution of the 2025 Nobel Prize-winning research by Mary E. Brunkow, Fred Ramsdell, and Shimon Sakaguchi for their discoveries concerning peripheral immune tolerance illustrates a fundamental shift in immunology, moving from a model of central control to a system of active, peripheral regulation. The pioneering era, challenging dogma, mid-1990s. Key figure, Shimon Sakaguchi. In the mid-1990s, the established view, known as central tolerance, held that the immune system learned to tolerate the body's own tissues primarily within the thymus, where self-reactive immune cells were simply eliminated. 1995, Sakaguchi's initial discovery. Working against this prevailing dogma, Shimon Sakaguchi identified a new, previously unrecognized population of T cells circulating in the body. He proposed these cells actively suppressed immune responses and prevented autoimmune attacks, suggesting that immune tolerance was not merely a passive process of cell elimination, but also an active, peripheral mechanism. He initially characterized these cells, which would later become known as regulatory T cells, Tregs, but his discovery was met with significant skepticism from the wider scientific community. The genetic connection, unraveling the mechanism, early 2000s. Key figures, Mary E. Brunkow and Fred Ramsdell. The next crucial piece of the puzzle came from genetic studies that established a molecular basis for the autoimmune suppressing function. 2001, Brunkow and Ramsdell's breakthrough. Mary E. Brunkow and Fred Ramsdell were independently studying the genetic basis of severe autoimmune diseases. They discovered that a particular mouse strain prone to severe autoimmune disorders had a mutation in a gene they named /BM, FOXP3, forkhead box P3. Critically, they also showed that mutations in the human equivalent of the /BM, FOXP3, gene cause a severe, rare autoimmune disorder in children called IPEX syndrome, immunodysregulation polyendocrinopathy enteropathy X-linked syndrome. This discovery provided compelling genetic evidence that the /BM, FOXP3, gene was essential for preventing the immune system from attacking the body's own organs. The unifying principle, connecting the dots, 2003. Key figure, Shimon Sakaguchi. The separate lines of inquiry, Sakaguchi's identification of a suppressive cell population and Brunkow and Ramsdell's discovery of the key regulatory gene, were then linked. 2003, Sakaguchi unifies the field. Building on the genetic findings, Shimon Sakaguchi definitively proved that the /BM, FOXP3, gene was the master control gene for the development and function of the very immune cells he had discovered years earlier. This crucial link established that /BM, FOXP3, expression dictates the formation of the regulatory T cells, Tregs, confirming their essential role as the security guards that maintain peripheral immune tolerance by actively monitoring and suppressing other self-reactive immune cells outside the thymus. The legacy, impact on medicine, post-2003 to 2025. The collective discoveries of Brunkow, Ramsdell, and Sakaguchi did not just answer a fundamental question in immunology, they launched a new field of research and created profound translational potential. Foundational shift. The findings transformed the understanding of how the immune system is regulated, proving that a dedicated, active system exists to prevent autoimmunity. Therapeutic potential. The discovery of Tregs and their master regulator, /BM, FOXP3, opened new avenues for medical treatment. The concept of manipulating Treg cells is now central to autoimmune disease treatment, developing therapies to boost Treg activity to suppress self-attack, e.g., in type 1 diabetes, lupus. Cancer immunotherapy, finding ways to inhibit Treg activity to allow the immune system to launch a stronger attack against tumors. Transplantation medicine, utilizing Tregs to prevent the rejection of transplanted organs. The 2025 Nobel Prize in Physiology or Medicine recognizes this decades-long evolution of discovery, from a theoretical proposal against the scientific tide to a universally accepted principle that is now underpinning a new generation of clinical trials and life-saving therapies.